The plastid which we are discussing in detail now is chloroplast. Let us talk about the scientists also. Plastids were discovered by as well as the term given by Haeckel. So Haeckel was the one who actually discovered these structures and called them plastid. Chloroplasts were observed by, observed means they were seen for the first time by Leeuwenhoek. And the term chloroplast is given by Skimper. They are dimorphic. The chloroplasts are dimorphic. That means they exist in two forms. Which are these two forms? One is granule and agranule. We will come to these two once we are done with the basic structure of chloroplast. Once we understand that, we will be able to differentiate between the granule and agranule, and we will also write in which type of plants we find these two types of chloroplasts. Chloroplast contains the green pigment chlorophyll. So the pigment which is found is chlorophyll. These chlorophyll are of various types, but mainly A and B. Plus there are other accessory pigments. Accessory means helping pigments. So chlorophyll will mainly participate in the photosynthetic reactions, but there are other pigments which need to help. So those accessory pigments are carotenoids, xanthophyll, phycobilins, phycoerythrin, there are many, but the predominant one is chlorophyll, A and B. In chloroplast, in the membrane of thylakoids, we will see the structure in a minute, these chlorophyll pigments are found in clusters. So these clusters are known as photosystems. Photosystems are group or cluster of chlorophyll. How many chlorophyll molecule? Normally 230 to 400 chlorophyll molecules make one group, which is known as photosystem. There are two photosystems which are present. One is called photosystem 1 and other is photosystem 2. The basic difference is the type of chlorophyll which is present in these two photosystems. Nowadays, we use the term photosystem. Earlier, this group of chlorophyll molecules was called quantosome. So this is, you can say, an old name, and photosystem is the latest or a newer name. So this is one which is more commonly talked about. Structurally, chloroplast is a double membrane structure. Let us first talk about, is there any other shape possible? So let us see the shapes of chloroplast. In higher plants, it is oval or little elliptical. Normally oval shape. So we can draw that shape. This is the shape in which we draw the chloroplast in higher plants. This is the most common shape. In case of a unicellular alga, that is Chlamydomonas, it is horseshoe shaped, which is also known as cup shape. So it can be written as cup shaped or horseshoe shaped. So the chloroplast which we draw is in the form of a cup shape or horseshoe shape, which is seen in case of Chlamydomonas. Another alga, Eulothrix, chloroplast is girdle shaped. So when we draw that chloroplast, girdle is basically C-shaped. So when you're seeing from the side, you just see one part and the nucleus is inside this girdle-like thing. So this is called girdle shaped. The filamentous green alga, Spirogyra, it is ribbon-like. So the chloroplast is in the form of a ribbon which is sort of folded. In these lower organisms, these are algae, chloroplast also contain some storage particles which are known as pyrenoids. They are not found in higher plants. So structure like pyrenoids, which are storage structures, and they contain a core of protein, a protein core which is surrounded by starch grains. This storage body is called pyrenoid, and these pyrenoids are embedded in the chloroplast of lower organisms. We don't find it in higher plants. So when we draw it, when we draw this chloroplast, we normally show these kind of pyrenoids also. The number of pyrenoids per chloroplast is variable depending upon the type. One more example, Zygeniма. In Zygenima, the chloroplast is star shaped, or it can be termed as branched. So the shape is, it may not exactly look like star shape, but it is branched. So that branched chloroplast is found in Zygenima. So these are some variation in the shapes of chloroplast. Talking about the number of chloroplast. Normally in lower organisms like Chlamydomonas and Eulothrix, there is one chloroplast per cell. Chlamydomonas is anyways a unicellular alga, it has only one. Eulothrix is in the form of a filament, but each cell works on its own, so it is considered as a unicellular structure. Again, there is only one. In case of Spirogyra, the number may vary from 5 to 6. So this one chloroplast in Chlamydomonas and Eulothrix, 5 to 6 in Spirogyra, and in general in plants the number is 10 to 20. This number is variable. The cells which are in dark, that is in shaded areas, they would have more number of chloroplast, and the cells which are exposed to direct light, they have less number of chloroplast. So this is again dependent on what is required by the cell at that time. If intensity is more, even less number of chloroplast can absorb that much of light with the help of chlorophyll. If it is in shade, you would need, or the plant would need, more number of chloroplast. So shape and number. Now coming to the structure. Double membrane, and we are talking about the chloroplast in higher plants. So we will draw the shape which is seen in case of higher plants, that is oval or little elliptical, double membrane. The outer membrane and inner membrane Both are made up of same protein and phospholipid structure, typical plasma membrane type of membranes. Outer membrane is more permeable, same as case of mitochondria's outer membrane. Inner membrane is rich in proteins and is selective in terms of the substances which can go through it, the inner membrane. Between the two membranes there is a space. This space is known as intermembrane space. The substance which is filled inside is rich in enzymes, ions and many more structures. This is known as stroma. Stroma has divalent ions, that is magnesium, manganese and ferrous ions. All divalent ions are present. In the matrix or stroma are also present ribosomes. These ribosomes which are present in plastids are 70S type, same as what is seen in mitochondria as well as in prokaryotes. Plastids are semi-autonomous organelle like mitochondria. So they have their own DNA which is circular and is known as plastidome. Plastidome. That is the name given to the circular DNA which is here. Plus, in the stroma are certain membranous structures. What is this membranous structure and how is this formed? I am going to erase it in a minute. I am just showing you how it is formed. When the plastid is young, when the chloroplast is being formed from the proplast, that is the precursor, then the inner membrane shows folds. These folds, they detach later on and they get arranged in the form of stacks. These stacks are known as grana. So how are these structures formed? They are formed by infoldings of the inner membrane. So outer membrane is smooth, inner membrane is folded in the young plastid or young chloroplast, but as it matures, these infolds they detach and form small structures which are known as thylakoids. So this single one which is here is a thylakoid. And there are many thylakoids. The number is again variable of thylakoids. It can be 10, 15, 12. Many thylakoids form a stack-like arrangement. This stack of thylakoid is known as a granum. So there are many grana. Let us draw a couple of these grana more. At some places, these grana are still connected with the inner membrane. Plus, these grana are also interconnected by membranes. These connecting membranes are known as stroma lamellae or frets. Frets or stroma lamellae, these connecting ones. So, at one end they could be connected to the inner membrane, at the other end to the granum, and between adjacent grana or between 2-3 grana also there are connections. The membrane of these thylakoids, thylakoid is this one single structure in the stack. In this are present the pigment molecules. The membrane of these thylakoids has the pigment. The green pigment which is present here is called chlorophyll. So chlorophyll is present in the thylakoid membrane, and thylakoids make stack-like structures. Circular DNA, that is the plastidome. Ribosomes, one more very important thing which is present in this stroma is an enzyme. There are many enzymes which are present here, but one very important enzyme is called rubisco. This enzyme is responsible for photosynthesis. This enzyme is the most abundant enzyme on the earth. Because every plant performs photosynthesis, every cell of every plant is almost green, the exposed part that we are talking of, and every cell has this rubisco, so most abundant. Other than these structures, the enzymes which are required for other processes like Calvin cycle, the dark reaction, so enzymes for Calvin cycle are also present in stroma. So structurally, chloroplast is a simple structure, double membrane. The inner membrane folds can be seen only in a younger chloroplast. After it has matured, we would find grana, and there are certain connecting structures which are stroma lamellae. They would connect the inner membrane to grana and granum to granum. So basic structure. Let us come back to this. We said that is they are dimorphic. There are two forms. Morph means forms. Two forms of chloroplast. Granule and agranule. Granule means grana present. What are grana? These stack-like structures are called grana. So if in a chloroplast these kind of structures are present, then it will be called granule chloroplast. These granule chloroplasts are present in C3 plants. C3 plants and C4 plants are discussed during the chapter of photosynthesis. C3 plants which are performing the normal photosynthesis, this is very, very helpful. Agranule means no grana. Where would we find chloroplast where grana are not required? This is seen in case of C4 plants, not all the cells. Some cells like the mesophyll cells of C4 plants, they have agranule. That means there are no grana. Not mesophyll cells, bundle sheath cells. Bundle sheath cells, they have chloroplast without grana. Reason, in these cells, light reaction does not take place, only dark reaction takes place. Grana have the pigment chlorophyll which is required for capturing of light, so they would be participating in light reaction. And stroma has all the enzymes which are required for Calvin cycle or dark reaction. So in these cells only dark reaction takes place. So there are two types, granule and agranule chloroplast. Coming to the functions of chloroplast, one very, very important function for which it is given so much significance It helps in photosynthesis. Now we can elaborate this thing as photosynthesis into many points. What are those points? It converts carbon dioxide into carbohydrates. It can be written as it fixes carbon. We can also write it as solar energy is converted into chemical energy, that is ATP. So photosynthesis, we can write many points in photosynthesis. One very, very important function. Second function is, during photosynthesis, oxygen is given out as a byproduct. Though it is a byproduct of this process, but molecular oxygen is required for all aerobic processes. So this molecular oxygen helps in all aerobic processes, whether it comes out of the plant, comes into the atmosphere, used by plants and animals separately, or it is used by the plant directly. So oxygen. One more function which is performed by chloroplast or plastids for that matter and mitochondria. So we are bringing mitochondria here also again. Chloroplast, or we can write plastids and mitochondria. Both are semi-autonomous. That means they have their protein synthesizing units, ribosomes, as well as DNA. So they are able to replicate on their own, and that is why they are known as semi-autonomous. If cytoplasm of a cell goes into a gamete, what all things would go in the cytoplasm? If it is a plant cell, chloroplast will also go and mitochondria will also go. If it is an animal cell, only mitochondria. So other than nuclear genetic material, is this genetic material not going into the cell or in the next generation? And it is in cytoplasm. So they help in cytoplasmic inheritance. Cytoplasmic inheritance is very, very important. So plastids, all plastids, because all plastids are semi-autonomous, and mitochondria, because mitochondria and these, they are all semi-autonomous organelle, they have their own genetic material. So this genetic material is transferred to the next generation through eggs, because eggs carry the cytoplasm, and that is why this transfer of genetic material is known as cytoplasmic inheritance. There are certain similarities which we find between chloroplast and mitochondria, and we also find similarities between chloroplast, mitochondria and prokaryotes. So let us compare these three things and see what all things are similar in those. Let us talk about the similarities now. Similarities between chloroplast and mitochondria first, and then we will compare it with prokaryotes also. Similarities between chloroplast, mitochondria and prokaryotes. And as we are talking about similarities, that means we are talking about the structures which are similar in all three. So in chloroplast and mitochondria first. Both are double membrane structures. Mitochondria is also double membrane. ribosomes which are present are 70S, 70S, again similar thing. Both have outer membrane smooth and inner membrane folded. In case of chloroplast, those folds disappear in mature. So I am writing only in young, because we would not see these folds of the inner membrane in a mature chloroplast. In case of this, outer smooth and inner folded. These folds in mitochondria are known as crista. So these crista are found in young as well as mature mitochondria, but they disappear in mature chloroplast. Both help in cytoplasmic inheritance. Same in case of mitochondria. The theory of their evolution is also same. We have already talked about the theory of evolution of mitochondria. Mitochondria are believed to have evolved by endosymbiotic theory. The endosymbiotic theory was that one prokaryote is taken in by a eukaryote as an endosymbiont. So if this is a larger cell which is a eukaryotic cell, and if in this eukaryotic cell lives a prokaryotic cell in symbiotic association, and this symbiotic prokaryotic cell is responsible only for one function. It could be respiration here, that is ATP generation, and it could be only for photosynthesis. So in that case, as evolution took place, probably this prokaryote later on became either mitochondria or chloroplast. So same is the theory for chloroplast. That means the larger eukaryotic cell had a prokaryotic symbiont. This prokaryotic symbiont was photosynthetic. That means it was performing photosynthesis on its own. It could have been a unicellular alga also. So if it was an alga performing photosynthesis inside a eukaryotic cell, and as evolution took place, this prokaryotic structure or cell now is known as chloroplast. So this is how evolution is taking place. There is evolution has taken place. There is one more proof to this. In dinoflagellates, dinoflagellates are photosynthetic protists. They have some endosymbiotic green algae. So in the body of a dinoflagellate, there is a green alga. What role this green alga must be producing or doing? This green alga is helping in photosynthesis. Dinoflagellate can perform photosynthesis on its own, but if it is not able to do that for whatever reason, this green alga would synthesize food. And what does the alga get in return? It gets protection from the dinoflagellate. So this also gives us a proof that probably these two structures, that is chloroplast and mitochondria, have also evolved by the same hypothesis, which is endosymbiotic theory. So many things similar in these two structures, and we also call both of them as semi-autonomous. We have not written that both of them have DNA and same things. Now if we compare this thing with prokaryotes, 70S ribosomes. This is the characteristic feature of chloroplast and mitochondria as well as prokaryotes. In mitochondria inner membrane, there is a protein called cardiolipin. We have talked about this when we were doing the structure of mitochondria. The inner membrane of mitochondria has a protein called cardiolipin. This cardiolipin is found or is present in the plasma membrane of prokaryotes. When we are writing this, let us add they have their own circular DNA. Here also circular DNA. One more similarity which we find between these two structures is a protein. Let me write it here. The protein porin, which is found in the outer membrane of chloroplast, is also found in the outer membrane of mitochondria. So porin in outer membrane, and here also same. So chloroplast and mitochondria share almost everything which is similar except for their major functions. One more point which they share is ATP synthesis. Mitochondria anyways we know is the site where ATP is synthesized by aerobic breakdown of sugars. In case of chloroplast, ATP is synthesized by a process which is known as chemiosmosis. A proton gradient is formed and which results into this ATP synthesis. So here also ATP is synthesized, here also ATP is synthesized. And if you compare these two structures with prokaryotes, there are two major things, 70S and the protein which is present. We can take one more similarity between these. Prokaryotes have a circular DNA, naked DNA, that means there is no envelope or cover around it. Same is the case with chloroplast and mitochondria. They also have their simple DNA which is not covered with any membrane, same as the case with prokaryotes. And because they share so much similarity, this theory of endosymbiotic origin is considered as a correct hypothesis on the basis of what we have in comparison. So this is just a similarity comparison between the three things, mitochondria, chloroplast first, and then these two compared with prokaryotes.